Hi, I'm Lewis Quayle, data scientist, bioinformatician and maintainer of lewisdoesdata.com, which is my website where I blog data science tutorials and guided data analyses. Today we're tackling differential expression testing using DSeq2 and adding gene level annotations. By the end you'll be equipped to identify differentially expressed genes and prepare your data for downstream analyses. So today we'll be continuing directly on from where we previously left off in the last tutorial. So all of the setup instructions for this tutorial were covered there. So if we get started, the libraries that we're going to load and the custom plot theme is exactly the same as those that we set up last time. So I'm just going to run those two code blocks without any explanation. And then because we'll be continuing on, we'll be reliant on using our DDS object from the last tutorial. So I've included this code in the companion blog post for anybody that didn't manage to set up the DDS object correctly. You can download the DDS backup RDS file from the link in the companion blog post and load that into your R environment and continue on with this tutorial from there. So the purpose of differential expression testing is to identify genes whose expression levels differ significantly between different experimental conditions, treatments or groups, which provides insight into the molecular mechanisms underlying the biological phenomena being studied. Now it's important at this stage to remember that DSeq2 requires untransformed raw counts as input for this analysis. So although we transform the data for QC in part 2, undertaking the variant stabilizing transformation in that case, the raw counts are essential for this step, and those raw counts are the ones which are stored within our DDS object. So we're going to use the DDS object from the last tutorial, and we're going to walk through the three main steps of the DSeq2 workflow, which are size factor estimation, dispersion estimation, and then model fitting with hypothesis testing. So the first stage is estimation of size factors, and before we look at the workflow using our DDS object, let's look a little more closely at the estimation of size factors using a demonstration dataset, because this relates to the library sizes that we saw in the last tutorial and how we can account for those differences in library sizes by creating scaling factors that normalize the raw read counts. So what we're going to do is we're going to create this demo dataset, this counts matrix, which contains counts for the 5 genes that I've listed here against samples 1 and 2. So if we run all of that and then we have a quick look at the demo data set, we can see that we've got a small counts matrix that will enable us to look in detail at how scaling factors are computed. So the size factors are scaling factors that normalize the raw read count data to account for the differences in sequencing depth and other technical biases across samples. So this is what we refer to as normalization. And this normalization step's crucial to ensure the comparison of gene expression levels between samples are accurate and not confounded by technical variability. So the size factors are calculated based on the median ratio of gene counts relative to a GeneWise pseudo-reference sample, the GeneWise geometric mean across all samples, that is, within DSeq2, which helps in achieving a comparable scale across all samples in the dataset. So there is a estimate size factors function that can be used to undertake this computation, but the following code chunk demonstrates the steps which are undertaken in the background. So the first thing is to calculate the row-wise geometric mean, which acts as a pseudo-reference. So that gives us the geometric mean for each of the 5 genes within our counts matrix. And then if we divide by the ratio of each sample to the reference, that gives us the ratios. We can then calculate the normalization factor for each sample by taking the median of those ratios. That gives us our normalization factors. And then what we do is we divide all of the counts within our demo dataset counts matrix by these normalization factors. So we can see now if we print the demo dataset, we get the raw counts, the unadjusted counts as we would have in our DDS dataset at present. And then if we look at the normalized data, we can see that those counts within our matrix have been adjusted to account for the difference in library sizes by dividing by our normalization factors. So that was just to demonstrate the step-by-step series of computations that happens when you run the estimate size factors function from DSeq2. And that allows us to normalize the data, which means that the gene-wise counts between samples are adjusted for the difference in technical variability, which ensures that any subsequent analyses are based on biologically relevant differences rather than technical artifacts. So luckily you don't have to remember all of those steps within the workflow. What we can do is we can estimate the size factors of the counts within our DDS object by passing the DDS object to the estimate size factors function, and then if we assign that back to the DDS object, we can see either by printing the summary of the DSeq dataset, we can see now there's a size factor stored within our metadata, size factor variable stored within our metadata. Or we can actually use the col data accessor function or size factors accessor function in order to actually view those size factors. Although, as we'll see later, you don't even need to explicitly run this step necessarily to perform differential expression testing in DSeq2. It has one major advantage in that it allows us to extract the normalized counts data. And I cover this in a little more detail in the accompanying blog post, so I'm not going to cover this here. But essentially, once you've estimated the size factors explicitly in the way that we've just seen, you can then use the counts function that we saw in the last tutorial to extract that normalized counts matrix by adding the additional argument that enables that. So after normalizing using the median of ratios method, the next step in the DSeq2 workflow is estimating the dispersion parameters for each gene in the RNA-Seq dataset. So dispersion measures the variability of gene expression levels across biological replicates beyond what would be expected from Poisson variation alone. DSeq2 uses a specific measure of dispersion that's related to both the mean and the variance of gene counts. Now, the dispersion estimation process in DSeq2 involves several steps, which is first of all computing the initial estimation of dispersions for each gene, which uses the maximum likelihood estimation method, or MLE. Given the count values of the replicates, the most likely estimate of dispersion is calculated, and then we fit a dispersion trend. So a trend is fitted to dispersion estimates as a function of the mean expression level. And this trend captures the overall relationship between gene expression levels and their variability, acknowledging that different genes have different scales of biological variability, but generally follow a predictable pattern. And then finally, the last step in the computation is shrinkage to the trend. So the initial dispersion estimates are then shrunk towards the fitted trend from stage 2, and this shrinkage step borrows strength across genes, resulting in more stable and reliable dispersion estimates, especially for genes with few replicates or low counts. And we can see these three steps when we run the conveniently named function in DSeq2, estimate dispersions. So similar to how we saw with the estimation of size factors, we simply pass the DDS object into the estimate dispersions function and assign the output back to DDS. And again, if we print the summary of the DDS object once that's finished, we can now see that the various different dispersion parameters have been added to this summary, so the different parameters that are computed during this process. Now, a dispersion plot, which can be made using some of the parameters that I've just spoken about, will show how the gene level dispersion estimates fit the model. You can create a fancy ggplot2 version if you wish, but DSeq has a function for this which is perfectly serviceable, which is what I've shown here. Now, running this function will give us a plot, and the model fit during dispersion estimation is displayed as a red line on the figure shown, which is a plot of the estimate for the expected dispersion values for genes at a given expression level. So each black dot is a gene with an associated mean expression level and maximum likelihood estimation of the dispersion, and then each blue dot is the final estimate following shrinkage to the fitted trend. Now, this plot, although it might seem complicated at first glance, is a great way of ensuring that your data are a good fit for the DSeq2 model, because we might expect that our data would generally scatter around the curve, with the dispersion decreasing with an increasing mean expression level. And if you see a cloud or different shapes here, then effectively that's a red flag, meaning that you might want to explore your data more to see if you have contamination, for example, high levels of mitochondrial genes due to dead or dying cells, or outlier samples. So essentially, even though we're now looking at the differential expression workflow, this is another quality control check that we can undertake. Once we've estimated the size factors and estimated dispersions, the next thing that you can apply to the DDS object is this n binom WALD test function. And again, we can assign that back to DDS. The WALD test evaluates the significance of coefficients in the fitted model. Specifically, it tests whether the log2 fold change estimate for each gene is significantly different from 0. And the WALD test statistic is calculated for each gene and a corresponding p-value is computed. And the p-value indicates the probability of observing a test statistic as extreme as or more extreme than the one calculated under the null hypothesis. Now let's just remind ourselves, the null hypothesis is that the gene is not differentially expressed, or there's a log2 fold change equal to 0. And the alternative hypothesis is that the gene is differentially expressed, or that the log2 fold change isn't equal to 0. So if the p-value is small, typically less than a chosen significance level, such as 0.05, the null hypothesis will be rejected, providing evidence that the gene is differentially expressed between conditions. So that's it in terms of all of the considerations for the DSeq workflow. Now, luckily, rather than having to remember these three functions and their sequential order, there is a convenient function in DSeq that will run all of these for us. So effectively, what we can do is, rather than running the last three functions that we've seen and assigning the outputs back to DDS, we can just run DESeq on the DDS object and assign that to, in this case, an object called DE. So I'm just going to show that it's not contingent on having run those by reloading my backup DDS object. And then I'm going to run that entire workflow and assign the output to this DE object. So I'll take a few seconds to run. And as you can see from the readout in the console, it's estimating the size factors, estimating the dispersions at a gene-wise level, and then the mean trend, and then fitting and then shrinking those estimates to the fit, and then fitting a general linear model and undertaking the test. So that might seem too good to be true. Now, in many cases, this very basic workflow and the steps that I'm going to show shortly might be good enough, but there are many different arguments that can be used to configure the behavior of the DSeq2 function, including using an alternative hypothesis test, which is the likelihood ratio test. A deep dive on these topics is outside the scope of this tutorial, so you can read more about that in the DSeq2 vignette if you're interested, and that's linked several times in the accompanying blog post. Now, if you were to print the DE, or the differential expression testing result object that we've just created, like so, you can see that in fact we still have a DSeq dataset. To access the differential expression testing result itself for each gene in our dataset, we actually have to call this results function on the output of the DSeq workflow. Now you can see that this function extracts a results table from the DSeq analysis, giving the base mean across samples, the log2 fold change between conditions, the standard error of that log2 fold change, and then test statistics such as p-values and adjusted p-values. And that's an important thing to note. So in DSeq2, the workflow applies multiple testing corrections to control the false discovery rate, or FDR, using the Benjamini-Hochberg procedure by default. So there is an argument to the results function that can be used to change this default behavior to different adjustment methods. Now note that all genes are reported in the output of the call to results, not just the significant ones. So that's going to become important a little later on. And you might have also noted when looking at this output that the information in the header here gives us some information about the two groups in our dataset that have been compared during the differential expression test to which the results pertain. So this is called a contrast. The contrasts that are available for testing will depend on the experimental design which was specified by the designer argument when we created the DDS-DSeq dataset object. So if you recall from tutorial 2, we used the DSeq dataset from matrix function call, and one of the arguments was called design, to which we passed a one-sided formula specifying the experimental design. And this design tells DSeq2 which variables in the sample metadata can be used to specify the groups of interest to compare and any interaction terms for differential expression analysis. So typically we'd decide the design for the analysis when we create the DSeq dataset object, but it can be accessed to check the design that's been specified and modified if required prior to differential expression analysis using the design function. So this design function where we pass in the DDS object will show us the design that we specified at the time that the object was created, and if we wanted to, we could pass in a different design. So we might say by rep, for example. I'm not going to run that, but that would allow us to pass in a different design. So in our case, we can see that we've indicated that we wish to draw contrasts based on the group variable in the experimental metadata. And because we've encoded this as a factor with 3 levels, CTR, IR and TGF, there are several potential contrasts that we could draw. For example, we could say we want to compare CTR versus IR, CTR versus TGF, or IR versus TGF. And this results names function can be used to indicate the contrasts or the model coefficients that can be accessed under the experimental design that we've specified. So in the analysis that we've undertaken, DSeq has automatically decided which member of our sample groups to use as our baseline condition. So in this case, as we can see from the output in the header, that's the CTR or control group. And the log2 fold changes in the results that are positive indicate that the respective gene is more highly expressed in the TGF group than in the CTR group. So in order to specify the contrast variable that we wish to use or change the directionality of the contrast, we can use the contrast arguments when we access the results using the results function, which is what I've shown here. So instead of just running results on the output of our differential expression testing pipeline, we can now specify the contrast argument with a vector of length 3, saying that we want to use the group variable and we want to use IR as our test condition and CTR as our base condition. So if we run that now, we can now see that we've got a different test condition and that CTR is still being used as our baseline condition. Now if we wanted to, as the example above shows, if we wanted to change the direction of that default contrast or that contrast, we could simply reverse the second 2 elements of the vector that we passed to the contrast argument. So all of these examples perform a differential expression analysis using a single named column in the metadata slot of the DDS object, but DSeq2 can perform much more complex analyses based on multiple experimental factors, which are so-called multi-factor designs, and these are actually very common in biological research. Now, one potential motivator for the use of such a design might be the discovery of a batch effect in the data, such as that shown in the simulated example in the accompanying blog post. So as I mentioned just before we looked at experimental designs, the output of the call to the results function reports the results for all genes in our counts matrix, not just the significant ones. In addition, the gene identifiers, in this case ensemble gene IDs, by which the rows are currently ordered, aren't very informative, and the format of the output, and more specifically the object class, isn't compatible with standard data manipulation tools such as tidyverse packages and functions. So let's start with the low-hanging fruit. So the results function that we've been using also has this tidy argument, which can be used to coerce the default DSeq results object returned to a tidy data frame, so that makes it tidyverse compatible, and then we can convert that to explicitly to a tibble. So if we run those in sequence, we can see first that we get this data frame, and then if we run the whole code chunk without the assignment, we can now see that we've converted it explicitly to a tibble. We will save that in our res object. so that we can start looking at adding annotation. So you'll notice that the output that I've just printed no longer shows a header detailing the contrast used, and the gene IDs are now a variable, which is named row, as opposed to being used as row names. And we can improve this output further by adding additional gene level identifiers or annotation and wrangling the data a little more to make the format more user friendly, such as ready for sharing with group members and collaborators. So the first thing to do would be to add some annotation. Now, Bioconductor, which is an ecosystem for bioinformatics within R, has a massive number of packages for annotation. And we're going to take a simple approach here of adding annotation using the annotation DBI and org-hsegdb packages. And the latter of these is one of several offline SQL-like database packages that exist in Bioconductor that are rebuilt every 6 months, which offer organism-level access to genome-wide annotation. So the first thing that we need to do is to load a package for that particular database. And the reason that we've done that now, as you can see from the output in the console, is because it has a function called select, which masks the default select function from dplyr, which would make some of our earlier data wrangling a little bit more verbose, and it means we're now going to have to call the select function from an explicit namespace so that we get the behavior that we expect. So once we've loaded this package, the first thing that we need to do is to identify the information that's available to us and which variables we want to add to our data, and we can do this by running this columns function on the org-hsegdb database object. And we can see from the output in the console, we get a vector of names of different annotations that we can, and information that we can add or access for this particular genome. To build our annotation data frame, what we need to do is to filter the database using a set of keys to extract the specific information that we want in relation to each gene within our results data frame. So what we're going to say is we're going to say, using the annotation DBI package and calling the select function on this human genome database, we're going to say the keys are the row names of DDS, which is ensemble gene IDs, as we can see if we just run that small chunk of code there. And we're specifically telling the function that the key type is ensemble. And then the final thing that we pass in is a vector of the columns that we want to access for this, which are the ones that we saw when we called columns previously. So we know that entree ID, symbol, and gene name are available within this particular database from the outputs down here. So we can say, matching the key types from our data to the keys within the database, extract the gene level information for which will add the entree ID, the symbol, the Hugo symbol, and the gene name. And we're going to save this in an object called annotdf. So we see in the console that an error has occurred. This is something that can be ignored. This occurs because there's often several different identifiers from other annotations used for each ensemble ID. And I say this can be ignored because we're going to deal with this issue shortly. If we want to preview the annotation data frame that we've just made, we can just run the head function and ask for the first 10 observations within that. And we can see now that we've effectively got a lookup table that has the information for the gene IDs that we have in our data, and we've added the matching entree ID, the matching Hugo symbol, and the matching gene name to each of those observations or ensemble gene names. So as I said earlier, we can almost ignore the fact that we've got a one-to-many mapping between the keys and columns in our data, because actually removing duplicate entries is fairly straightforward. We can actually just use the code showed here to determine the number of duplicate entries. So what we're doing is we're saying within this variable, within our data frame that we've just created, so a vector of ensemble IDs, return a Boolean vector, whether there are duplicates that exist or not, and then sum up the number of entries which evaluate to true. So we can see, having done that we have 3,992 duplicate entries within our ANOT-DF object, which is 67,233 observations long, so a relatively small proportion. And in order to remove those, we can actually call distinct on the data frame with respect to the ensemble variable, and we can just add this .keepAll equals true argument, which means keep all of the other variables when determining the unique entries for the variable within that data frame. So what we can do is we can assign that back to an object that now is our annotation data frame with all of the duplicates removed. And we can see that we've now got this object here, which is 63,241 observations versus our input data frame, which was 67,233 observations. And if you want to explicitly check whether you've removed the number of duplicates that you'd be expecting, you could quickly perform the subtraction in the console and see that we get the same difference in the number of observations as there are the number of duplicate entries in the input data frame. So now all that's left to do before we're ready to save the results is to join this information onto our results data frame and then do a little more data wrangling to make the final output as user-friendly as possible. So after that, we can then easily determine the number of significant results at a specific significance cutoff value and then filter the clean results data frame to return the genes with a significant FDR adjusted p-value. So it's important to note at this stage that some people will also want to incorporate a log2 fold change cutoff here. It's actually more desirable from a statistical standpoint in terms of correctly controlling the type 1 error rate to incorporate this threshold into the hypothesis test and then filter for significant results. And it's more correct to do that than it is to apply this log2 fold change threshold as a post hoc filtration step. So this is exactly what I'm going to show briefly before we undertake any downstream data wrangling. So I've got one final call to our results function that we've seen previously, and we're passing in our differential expression testing result from the DSeq2 workflow. We're using the contrast that we specified earlier, where we're saying our variable of interest is group and we want to test for different genes differentially expressed between our control baseline condition and our IR or irradiated group. We want to return a tidy data frame as the output, and this argument here allows us to apply our log fold change threshold. So we've said flag any genes that have a log2 fold change greater than 1, and the alternative hypothesis argument here allows us to say whether that's in a negative, positive, or either direction. So in this case, I'm saying I want genes that have a log2 fold change of either greater than +1 or less than -1 to be called as differentially expressed. And then I'm specifically saying use the Benjamini-Hochberg adjustment method and then coercing that to a tibble. So if we run that, we can see, just as last time, we've got a tibble and we can see the head of the results. And we're now in a position where we can start to wrangle the results and add on the annotation that we've just created. So if I scroll down to my wrangling code, what we're doing is we're passing the results object that we've just created to the left join function, and we're saying join onto these results the annotation data frame where we've removed the duplicates, and we're using the row variable as the key, and we're matching that to the ensemble variable within this data frame because those are the common keys between the two tables. So that will actually enable us to undertake the join. So if we just run those two rows, we can see that now. So we've joined on the annotation information to the differential expression testing result using the ensemble ID as the common values on which to make that join. And then what we're going to do is we're going to tidy up the variable names by running janitor clean names. So that coerces the variable names to syntactically correct variable names, all in snake case. And then what I'm doing is I'm just moving a few of the variables. So I'm moving the entree ID symbol and gene name variables to after this variable, which is currently called row. So that's what this line does. So if we run all of that, we can see that. So we've got the same output, but now these three columns, rather than being at the end of the data frame, are now in variables 2, 3 and 4. And then what I'm doing is I'm renaming a few of these columns. So I'm going to rename the row column to ensemble. I'm going to rename entree ID to simply entree, and I'm going to rename log2 fold underscore change to simply LFC. So that will tidy the data frame up quite nicely, and it means that not only are our variable names syntactically correct, but they're also not excessively verbose when it comes to typing them later on for making visualizations, for example. And then finally, we can apply a filtration step to remove any of the adjusted p-values that are NA, and then we can arrange the output according to the adjusted p-value. So finally, if we wanted to count the number of significant results that we have in our data frame, we can use the dplyr's count function, passing in our data frame, and then I've created a variable to count on, which is where we're testing to say, is the adjusted p-value less than 0.05, and I've given that the output count, the number of instances of that, the name count. So we're first going to need to actually make the assignment, because I forgot when I ran the last few lines of code, we didn't assign the output back to the resclean object. So if we do that first, and then when we run the count function call, we can now see that we've got 238 differentially expressed genes within our data set at an FDR adjusted p-value of less than 0.05, which is differential expression between the irradiated and control groups above a log2 fold change threshold of 1, which is an absolute log2 fold change of 2 or more in either direction. So if we just want to filter to the differentially expressed genes with p-values less than a given threshold, such as 0.05, we can do that using the filter function, and we can assign that to an object in our environment if we wanted to save that, for example. So if we print that object we've just created, we can now see that we've got a data frame with exactly the same number of variables, but we've only got 238 observations, which is equivalent to the number of differentially expressed genes as we've just seen. So finally, if we wanted to write out those results, we could use the write_csv function from the dplyr package, passing in the object that you want to write out, and then the file path, and in this case I'm using the here function to build up a file path relative to my project top level. So I'm going to save these results in the results table subdirectory of the results directory within our project, within our R project, and I'm going to name that file IR versus CTR all, to signify that it's not just the significantly expressed genes, but it's the entire differential expression testing result. That's the end of today's tutorial. I hope that was useful, and I'll see you in the next one for how to visualize these differential expression testing results.